So really the methods of social neuroscience are borrowed from elsewhere. So we've got our kind of classic psychological methods, which I won't really go into, so response time measures, questionnaire measures, and so on. And the neuroscience methods, which I'll focus on, so I'm guessing that you have different levels of background and exposure to these methods. So for some of you, this is a recap. For some, it might be relatively new. For those who it's relatively new, it is worthwhile spending time outside the lecture reading this, okay? So the methods of cognitive neuroscience can kind of be divided according to their temporal resolution. So their ability to detect things happening in time, and their spatial resolution, their ability to detect things on a large scale or a small scale. So here, this is our kind of spatial resolution, temporal resolution. So something like fMRI, for instance, happens in the order of tens of seconds, maybe kind of, well, yeah, tens of seconds is seen as being the temporal resolution. So that's pretty slow in cognitive terms. So fMRI is kind of sluggish, and it operates at this kind of level here, which is about a millimeter level, for instance, there. I won't go through all of these. Whereas if we take something such as this, so single cell recording is an invasive method where you put an electrode inside the brain, and you're then recording from single neurons. So now you're down at the neural level. And here you can get things happening at millisecond levels, upwards in effect. But here you're measuring from single neurons. So fMRI, you're not measuring from single neurons, you're measuring kind of millimeter to centimeter kind of level. Okay. So the different methods have their different kinds of advantages and disadvantages. And they're also based on different properties. So EEG, for instance, is based on electrical signals emanating from your head. So whilst you're thinking, writing, or whatever, you have little electrical signals and magnetic signals coming out of your head. And this is effectively what EEG is doing. It's a recording method, by which I mean that it's not actually stimulating the brain, it's just recording what is coming out of the brain. So in that sense, it is safe, and it's also non-invasive. Whereas if we take something like fMRI, this is also a recording method, non-invasive, but here it's not actually measuring electrical activity, it's actually measuring something to do with the blood supply, and I'll talk about that. So you've got these different measurements. And also you've got some methods that are stimulating the brain as well. So TMS, which I'll show you a little movie clip about, this isn't just recording what's happening in the brain, it's actually affecting the brain itself. So other kind of stimulation methods would be kind of psychopharmacology, so taking some kind of substance and then measuring effects on behavior. That would be a stimulation method, albeit chemically, and so on. Okay. So this is kind of the landscape in which these methods operate. Within one kind of division as well is whether or not you're measuring the nervous system in terms of the central nervous system, typically the brain, or the peripheral nervous system. So there are some methods in social neuroscience that are based on measuring the peripheral nervous system with the idea that this is reflecting things that are happening in the brain. And one advantage of measuring often the peripheral nervous system is that it's often tapping kind of unconscious social process. So things like sweating would be a classic kind of measure of the peripheral nervous system, where you might kind of be having a fear response and you ask them to fill in a questionnaire and they say, I'm not scared at all. And here, they're not necessarily lying. It's just that their body's giving a somewhat different signal to their own kind of reflective state on this. So the peripheral nervous system is obviously kind of the eyes, the body, and so on. So again, measuring heart rate, measuring respiration, and so on. These are measures of the peripheral nervous system, but with the general assumption that they're being influenced by something centrally. So this is our skin conductance response here. So the interesting thing about that is that it can occur in the absence of even a conscious perception of a stimulus. So here, this would be a fearful face presented for 20 milliseconds. You ask the person, what did you see? They'll say nothing, okay? But you can show that they have this. So this is kind of an interesting part of the toolkit, very different from, say, a questionnaire measure where you're asking people to reflect on things, okay? And the other measure that's been looked at here is EMG. So this is looking at muscle activity. It's actually electrical signaling in the muscle. So you have electrodes attached to the face. So the kinds of things that are done in social neuroscience is that you have, say, pairs of electrodes here. These are your frowning muscles, and kind of here, your eyes are kind of your smiling muscles, believe it or not. It's not all in your mouth. What you might do there is that, for instance, there are examples of people, again, kind of interacting with racial in-groups and out-groups. They fill in a questionnaire saying, oh, yeah, this person was delightful, but you can show that they're frowning a lot more when they're interacting with the out-group than the in-group. That doesn't mean necessarily that they're lying, but it does kind of pose questions, you know, what's going on, which measure do we believe, and so on, you know. So here, these are kind of measures now typically that are involved in the central nervous system rather than periphery. So as you know, neurons communicate with each other electrically as well as chemically, and there are various ways of measuring the, in effect, the electrical activity of neurons. One at the level of individual neurons or kind of clusters of neurons together, and one at the scalp, which effectively means you're measuring millions of neurons that happen to be firing in sync. So you've got very low spatial resolution here if you're measuring at the scalp, but you've still got your good temporal resolution. As soon as neurons are firing, in effect, you can measure it, both at the scalp and in terms of sticking an electrode in. So here, these are single-cell recordings. So they're typically done on primates, but they're also done on humans when they're having brain surgery. And they're wide awake. So they have some clinics where they literally are just sat reading a magazine with these electrodes in there, just recording, trying to find out where their epilepsy is, but serving other functions as well. So, you know, it's not something that is necessarily done under surgery. Obviously, it's put in during surgery, but the human or the animal can have these implanted for quite a while. And here, what you're actually measuring is the firing of individual neurons generating action potential. So again, here, if we think about mirror neurons, the measure there is how many action potentials is that neuron producing a second in response to the animal seeing somebody pick up a peanut, or in response to a bar of light, or something like this. And obviously you compare that against spontaneous activity, how much that neuron fires when it's not doing anything, or how much that neuron fires when it's not doing something else. So this is a fun example of the so-called Jennifer Aniston neuron. I don't know if anybody has come across this. This is from humans in Los Angeles, of all places. It's, where, in effect, what they do is that these are neurons actually in the hippocampus, so part of the brain that's normally seen as being specialized for memory rather than perception. And the hippocampus is also often a site of epilepsy as well, which is why a lot of the information that we have comes from there. And you can present photos on a computer screen to a human participant, and you just then record from a set of neurons, and eventually you'll find a neuron that goes when it sees Jennifer Aniston. The researcher says, oh, that's interesting. It types in Google images for Jennifer Aniston and pulls up images, and then sees whether it responds to other images of Jennifer Aniston. This is how it works. So all these images here, and I'm sorry you can't see them, it's Jennifer Aniston. These are kind of the bar charts. So these are the number of action potentials over time there. So in response to Jennifer, this is one person, one neuron, but obviously they've done it lots of other times. It doesn't respond to Jennifer Aniston when her ex-husband Brad Pitt is in the photo. It does respond to Lisa Kudrow, who's also in Friends as well, this particular neuron. These plots there, it just kind of shades them in, the blue ones here, just shades in whenever neuron produces an action potential, and then you repeat the image over time, you kind of build up a map as to how it responds. Where do these come from? Well, obviously, they're probably learnt. You probably weren't born with a Jennifer Aniston neuron. You just learned to associate the features from different viewpoints and things there. There's Luke Skywalker, Darth Vader, Yoda. They're all in your hippocampus, and this is kind of the stimuli that they search on. Also, as they come to know the doctors in the hospital, they start to find neurons that respond to the doctors as well. So when the doctor walks past, they can see on the kind of oscilloscope thing the neuron is kind of going. So kind of very intriguing, enables you to actually build up a detailed picture from individual neurons as to how things like faces and people are coded in the brain. So obviously one problem with that method is that it requires on patients undergoing surgery. Their brains might not be normal anyway if they're undergoing surgery, so they're an atypical population. And it also limits the number of labs that can do that kind of research. EEG, on the other hand, is something that has a long history. It was developed in the 1930s, used mainly for looking at kind of sleep-wake cycles and so on, but can be used to address more cognitive questions. So here, this is somebody in a fetching EEG cap. And again, this isn't actually stimulating his brain with electricity or anything like that. It's just recording the electricity that's being given off. So when you're engaged in a thought, or let's stick with the example of seeing a face. So when you're seeing a face, the neurons in your brain that respond to faces more than to other stimuli will all fire, and they will all tend to fire in synchrony. And these give off a larger electrical current that will then effectively travel through the skull to the scalp. The problem is that even when you're seeing a face, all the other neurons are probably generating some background spontaneous activity over time. So in the example of showing a face and seeing the electrical activity given off, you just see nothing. You just see pure noise. And that's kind of the old EEG signal that they used to look at, see whether people were awake or asleep or whatever. So what you can do is that you show a face several times or different faces, and you effectively average together this. And the idea is that all this kind of background chirping of neurons and so on will effectively cancel each other out the more trials you give to a person. So you're left with just that signature, which is involved over time, that always responds to faces, for instance. So here, this is time. Here, this is to a sound. It doesn't really matter what it is. But this is just with one trial, 10 trials, 50, 100. So as you kind of average across trials, you get rid of this kind of background kind of fluctuating electrical activity, and you're left with something that's called an event-related potential. And what it looks like is a series of negative and positive peaks. What do the negative positive peaks mean? In themselves, they don't really mean anything. So the fact that they're negative or positive doesn't matter. All it means is that if you've got neurons kind of aligned this way, you might have a flow of electricity that way, which means that you've got positive here, negative there, depending on what it is. So it just depends on the orientation and how they are in the brain as to whether or not they're negative or positive. The way that you might kind of use them cognitively is to kind of then kind of take a peek and see what kind of experimental manipulations matter. So this is an example again for faces. So if you show somebody a face, you get this large negative peak at 170 milliseconds that's called the N170. You don't get it here with the green line, which is for objects. But interestingly, you do get it when you see a dog's face as well as a human's face. So it's coding something that is kind of non-specific here. So again, that's a kind of a cognitive question there. Is it just responding to humans, or is it responding to other kinds of faces? And it is responding to other kinds of faces. Does it respond to other, to out-groups more than in-groups? There's kind of a bit of a literature on this as well. Not much. It doesn't make that much of a difference. So you can effectively use this as a signature in which to kind of explore other questions in the cognitive domain. It responds to smiley face as well, so 2 dots and a curve. So again, it's some kind of abstract coding, but not other objects. So EEG has a high temporal resolution, which means that as soon as neurons fire, you can detect it at the scalp. But it has a poor spatial resolution. You can't necessarily tell from the signature at the scalp where it's coming from. And also, there are some regions of the brain where all the neurons aren't aligned with each other, or they kind of aligned in such a way to cancel each other out. So you can't detect EEG signals from the hippocampus. So, again, these key regions, just because of the anatomy as to how the neurons are laid out. You need all the neurons to be laid out in parallel, which is typically the case in the cortex. Actually, if you think of it as a long sheet, all the neurons are kind of aligned next to each other like this. So it's good for things like the cortex, but not the subcortex. before I go into fMRI, any kind of questions about those methods? I am rattling through them. Okay. So brain imaging actually isn't, or functional imaging, which is mainly fMRI these days, is not measuring the activity of neurons. Okay? And this is really important to know, and it's kind of led to all kinds of, I wouldn't say skepticism, but it's certainly a more complicated story in terms of what we are measuring in fMRI than it is with EEG or single cell recordings. Okay? What we're actually measuring is something related to blood flow, which is why it's called a hemodynamic method. And the basic kind of story as to how fMRI works goes like this. And that's that when neurons are generating electrical signals, they are consuming oxygen. The oxygen they need in order to fire comes from the blood supply. So you've effectively got this chain reaction of chemical signaling that when a neuron is firing a lot, it then signals to the blood supply, give me more oxygen. The blood supply then responds by giving it more oxygen, in effect. So the blood vessels, which are the capillaries, effectively dilate as neurons fire. But this doesn't happen in real time. It happens several seconds after a neuron has been firing, okay? So fMRI's got a not bad temporal resolution, but you're not observing the brain in real time because it takes that sluggishness for that to happen, okay? The thing that you're actually measuring here, so PET is a forerunner of this, and what you used to do in PET, and you still can do, is that you inject people with radioactivity, so radioactive water or something like this, and what you effectively measure is how that travels around in the brain. So if the blood vessels are dilating, you will see an increase in radioactivity in that part of the brain, okay? One of the good things about PET is that you can actually then radio label other isotopes, so things like dopamine agonists or serotonin agonists, and then you can see how those are kind of selectively uptaken or binding. So fMRI was seen as being an advantage of PET in that it doesn't use radioactivity, so it's a lot safer. And the way that it works, in effect, is that rather than having a contrast agent, so the contrast agent is adding radioactivity, you can use the brain's own natural tendency for deoxyhemoglobin to change its properties when it is converted from oxygenated to deoxygenated. So PET is literally measuring the amount of blood in a region. fMRI is actually measuring something even more complicated than that. It's measuring this transition between oxy and deoxyhemoglobin as a result of this kind of cascade of processes. And that's what it looks like if you put your head in a scanner. So if you do want to take part in an imaging study, if you go, our scanner on campus is at CISC, C-I-S-C, the Clinical Imaging Sciences Centre, and they're always looking for healthy volunteers. You'll get a picture of your brain or a movie of your brain if you want that. Has anybody been there and had a scan or somewhere else? Yep, okay, so a few of you have. Okay. So this is the kind of the thing that you're measuring in fMRI. So we've got time across here. So you've got to imagine at 0 seconds you've got the firing of neurons. What happens when you fire neurons is that you've got a conversion from oxyhemoglobin to deoxyhemoglobin. The neurons are saying, give me oxygen. So you've got this conversion from oxy to deoxyhemoglobin, and this is what you see here in this little dip. Then what happens is the blood vessels respond and they kind of open up and they pump a lot more oxygenated hemoglobin in there, and this is effectively the signal that you're measuring. So although neurons are using oxygen, you paradoxically get more oxygen in the region by being active. So you kind of imagine if you're using oxygen, you've got less oxygen, but because you overcompensate, it goes the other direction from what you might imagine. So it's called overcompensation, and this is effectively what's happening in fMRI. But notice that, and then eventually it kind of overcompensates the other way, and then it goes back to normal. So this is kind of, depending on the scanning sequence, a kind of a 16-second process for it to go back to normal. So this is quite slow in terms of cognitive terms. And effectively what you're doing in an fMRI experiment is that you might, for instance, show a person a face versus an object, and you're asking the question, where has a large bold response to a face relative to the other? So you imagine if you're presenting that this is your face and you've got another line that's an object, it might look something like that, where you've got a low kind of bold response. And in effect, what you're doing is that you're doing a t-test at that little point in the brain, saying, is there more kind of oxygen there when it's looking at a face than when it's looking at an object? And what you're doing statistically is you're effectively doing a gazillion t-tests at every part of the brain. Should it be significant, you colour that part of the brain in. So these kind of nice images that you see of the brain, they're not the firing of neurons. In fact, what they are just maps of statistical significance, just by colouring in the size of this response relative to others. And it always has to be relative to something. So you've got to remember that the brain is consuming oxygen all the time. If it isn't, you're in trouble. You're having a stroke or something bad is happening to you. So you have, and different brain regions consume different levels of oxygen to others. So some brain regions require more oxygen than others for reasons that isn't fully understood. So you always have to have some kind of baseline in which to kind of understand. So you don't just put somebody in a scanner and figure out what they're thinking. You need an experimental design, you need control conditions, and so on. So here you might imagine that if it takes 16 seconds to, for this to kind of peak and then come back down, that you've got the most boring experiment you've ever done in your life waiting for the next stimuli. Fortunately, what happens is that these different responses can effectively summate, so you can add them together. So if you've got three faces all in one, you will have that bold response times 3, pretty much linearly. So here, what you've got to imagine is that this is time, and these are stimuli that you're presenting to the participant. Maybe in between you've got, houses or other objects. So what you're effectively doing is that this is the stimuli you're presenting. This is how you know your brain responds to those stimuli. You're effectively superimposing that on each of these time points. And what you're saying is, this is what I predict my data will look like if it's a face-responsive part of the brain. Okay? So it's not quite as simple as comparing this against another one, unless you've got 16 seconds between your stimuli. If you kind of put your stimuli together like this, this is what you're doing. And effectively, you're just doing a kind of a regression or a correlation. Ultimately, you're kind of saying what brain region produces a signal that looks like that. Okay, and based on your experimental design of how you're presenting your material. So it becomes more complex. Okay. So again, in order to figure out whether a brain region is active, you always need some kind of baseline or some good control condition. So again, there's some real problems in some literature in that if you haven't got a good control condition, it looks like that one part of the brain is kind of active. So in this example here, if you compare faces against, say, a fixation cross, and then you conclude that that part of the brain is specialized for faces, that would be an erroneous conclusion. Maybe it's specialized for big things relative to small things, for instance. Or similarly, if you've got faces that are in color, but then your stimuli that are objects happen to be black and white, you would draw the incorrect inference that you've got a face region of the brain, but actually, because you've not thought about your stimuli carefully enough, you might be looking at parts of the brain sensitive to color. And believe it or not, you would think nobody would ever make that mistake. I promise you, there are so many things out there in the literature that, or often there's a trade-off. There's never a perfect control. So it's a question of what are you willing to sacrifice. Do you always have to check that your stimuli are matched for luminance, for instance? Not necessarily, but if you're interested in the back of the brain, you might certainly want to think about that. So again, these are some of the problems that you kind of have with brain imaging. Yeah.
Maybe you can explain it, but how are they mapped onto an actual brain? Is
Yes, no, that's a good question. So you have what are called voxels. So if you think about a computer screen made up of pixels, a voxel is a kind of a volume element. So you divide the brain up into, typically it's 3 by 3 by 3 millimeters cubed. And you do that lots of times. So you actually end up with, I've forgotten the number, maybe hundreds of thousands of of voxels that you're kind of testing. So one of the basic problems that then generates is that if you're doing statistical comparisons at each and every voxel, and you apply something like p is less than 0.05, you will have thousands of brain regions that are active just by chance. So there are various ways in which you can deal with that. So one way is obviously you've got a hypothesis that that's the region which I'm expecting. So therefore, you're effectively just doing one t-test or one statistical test rather than a gazillion on each and every voxel independently. But then, of course, you don't generate new hypotheses unless you're looking for the whole brain. But this is one of the ways in which that's kind of done. It's called smoothing, which sounds really dodgy, but actually it's fine. It's normally done in the literature. There are some examples in which you don't smooth your data. One of the things that you would do here is that, in effect, you put what's called a Gaussian distribution on it, or normal distribution. In effect, you're spreading the activity on that voxel out to its neighbours according to how far away it is. So what it would mean in this case is that that voxel there, which doesn't appear to be active in its own right, after smoothing, it would get activity from its neighbours that would actually switch it on. Whereas this voxel here, that looks to be active but doesn't have any neighbours, would in effect be switched off. So one of the things you're doing here is that you're identifying large clusters and switching off small clusters. And that's one of the ways in which you get around the problem of lots of little regions being switched on by chance. If that's the case, the idea is it would look more like a little mosaic like this than a kind of a larger contiguous region. But of course, if the brain were to be made up of a mosaic of different responding things that aren't adjacent to each other, smoothing becomes a problem. The other reason why you smooth is that everybody's brain is anatomically a bit different. So it might be that in my brain, my face area is there, and in your brain, your face area is there. And by smoothing, you kind of, in effect, you kind of blur the boundaries between these regions, and it makes it a higher probability of finding that region. It might mean that that region isn't representative of well, it's kind of representative of the group, but it might not represent well any of the individuals in that group. So there are some people who look at it more at the individual level rather than the group level, or effectively run a series of case studies on normal people rather than this. Again, they're kind of subtle, but it's really crucial because it really affects the results you have in effect. So again, here, this is one of the kind of biggest controversies since the birth of social neuroscience, and it was about a paper that had what was called voodoo correlations in the title. And people who in the social neuroscience literature got very upset, and they wrote the editor, and the term voodoo was taken out, but it still appears on the author's website, and everybody calls them voodoo correlations. But basically, the problem here was about linking fMRI data with questionnaire measures. So if I were to give you a questionnaire measure, and then I bring you back a week later and give you the same questionnaire measure, your results would probably correlate with themselves about 0.7, for instance. Okay? So you're not going to give exactly the same answers, but they'll be broadly similar. If I were to put you in an MRI scanner and get you to do a simple task, like moving your hands or seeing a face, then I get you back a week later and I compare the pattern you get a correlation there of, again, about 0.7, 0.8. What we have here is a whole number of studies in the social neuroscience literature, and this is just a frequency distribution of the correlations reported in these papers, often in high profile. So remember, most questionnaires have test-retest of 0.7, and fMRI does as well. So the question here is, how do you get correlations in the literature that are 0.8 or 0.9? Okay? And this led to a whole host of problems. So basically, that these are, in effect, type 1 errors, significant results that shouldn't be significant results. And it was kind of acrimonious. So people were going to job interview. And the other thing was acrimonious. So each one of these is a numbered study. So some people would be consistently labelled in the red, and they would be going to a job interview at prestigious universities, and they would get wind of the fact that their 3 papers were in the red zone of this histogram. So it's also kind of very kind of politicized as well. So some of the people in this red zone were arguing, actually, there's a whole other distribution on this side, but these papers never made it into publication because of the publication bias, for instance. That would be one thing. So actually, it is about centered on 0.6, which is where you would expect this distribution to be centered based on the no test, retest reliability of this. So that was one argument of it. There's also been various other statistical arguments as well in terms of what they're doing is that they're looking at a set of voxels. What you would do is that you look at a set of voxels that are significant, then you go in and choose a voxel and you report the correlation of that voxel. So here you're effectively doing what's called double dipping. You're choosing something that's significant and then looking within that to find something that's significant even more. So you've got 2 kind of things, and it just boosts what appears to be a correlation. Anyway, it's kind of led to a rather fierce debate about how to deal with this, and it's kind of given social neuroscience a bit of a bad name that I don't think it deserves, but certainly when you're reading papers, you want to look how impressive their correlations are. So now nobody wants a correlation above 0.8 in their paper, whereas before, you'd be writing to the editor of Nature or Science with your correlation of 0.9 or, nearly 1 here, which you just don't see in behavioural sciences. So again, I don't want to make a big point about this, but with fMRI, what you see is kind of what you get in. It depends how you analyse it. You're not seeing the activity of neurons, and you are making statistical assumptions. So it's not the same as looking at, for instance, the genetic code where you're not making an error. There is no error to be made unless your equipment is faulty. Here, your equipment might be working fine, but you may get something out that is not what you expect with this. Any questions about voodoo? Okay, right. Yeah.
I just wanted to reiterate my question about the previous slide.
Yeah.
Because I just didn't. No, previous again.
Yes.
If you were to actually, because you said those voxels, they have neighbours, right? And the neighbours represent actual neuronal neighbours.
Yes. Yeah. Each voxel will have lots of neurons in it, but yeah.
That is a spatial map of the brain.
Yes. Yes.
If you were to draw a brain on the overlapping that, how would it look? What orientation would it look?
Oh, I see. Right, no, you've effectively got slices within it, so this would be a 2D slice, so this is how it's done. You've got slices. Oh, I see how they're oriented. My understanding is that the statistics don't really care about that. It's just how you display it. It's kind of irrelevant. You always tend to scan like this. That's how the scan is from top to bottom, in effect.
So that would be like looking down from a bird's eye view of the neuron?
It doesn't matter. It doesn't matter in a way, because you could present it on the outside, you could present it as slices. It does it in 3D, so it will take in anything that's next to it. The algorithm doesn't care whether it's in cortex or non-cortex or whatever.
Okay.
Yeah.
Excuse
me. Yeah.
Because on the next slide, sorry.
Yeah.
The voodoo correlation. The next.
Yeah.
Okay, because I think that some of the early literature confounded by non-independent analysis, but I'm a bit confused. So, in what kind of sense are they non-independent?
Yeah, so the kind of thing that would be done is that you take a set of voxels that are shown to be active, and then they would report a correlation between one of those voxels and the measure. And often it's the most active voxel, or the one from that. So in effect, you've already taken something that is significant, and then you're then choosing which voxel to present in your graph, and you draw the correlation on that. But yes, you're right, and some people have kind of argued that they didn't do this there. So some people say, no, we didn't do that, despite you thinking that that's what we did. Yeah.
But isn't that an issue throughout social science?
Absolutely, yes. No, absolutely. It is, yeah.
To
It is not unique, and I think that that's why one of the unfortunate things that's kind of given it a bit of a bad name is that it's nothing to do with social neuroscience or whatever. It's a purely statistical thing. I guess it's just a bit of a wider. It's hard to say myth because even the top journals kind of understand this, but. I guess there's kind of a bit of a naive sense that these sorts of things really are the true workings of the brain rather than some hypothesis-driven statistical map or whatever, And I think that that's kind of where the debate lies. Okay. Right, so in terms of... so those are, in effect, recording methods where you're recording activity in the brain. There are also ways of manipulating brain activity or stimulation methods. So classical kind of neuropsychology is, you wouldn't really call it a stimulation method, but you are manipulating the brain. So these would be patients with amnesia, patient with a stroke to the left superior temporal sulcus or whatever, where they effectively, you have manipulated the brain by accidents of nature in which those neurons are dead, they've had a stroke there or some other brain damage, and in effect you have a look what the profile of these patients are in order to figure out what the building blocks of cognition are, to kind of address questions like, what is the social brain? Can you damage this part of the brain only affects social cognition and nothing else? Those are the kinds of questions you can ask using this method there, whereas fMRI is often seen as a correlational method. You're kind of correlating brain activity with behaviour. Here you're manipulating the brain and then kind of observing the consequences of that manipulation. It's kind of almost the other way round. And there are various ways of doing it. So this is kind of classic neuropsychology where you've got two patients. You could have two groups of patients with lesions there or whatever. I'm not really going to go into that. So 0 means that you're average. So these are Z-scores, how far away you are from the mean. So patient NK is very good at recognizing fear, but really bad at recognizing disgust, whereas patient SE shows the opposite profile. So the kinds of conclusion that's been drawn here is that basically there are different neural resources in the brain for the processing of fear versus the processing of disgust, and that you can lesion one without lesioning the other. And in fact, you don't even need to know where the lesions are in order to draw that inference, although that would be another step beyond that to do that. Now, of course, it might not be the case. You would obviously want to test them on a whole range of other things. So, for instance, one claim made with fear is that the eyes are very important for recognizing fear. So if you don't draw your attention to the eyes, maybe you're going to be down with fear, but actually it does mean you've got a fear deficit. So again, you have to kind of explore these other interpretations. But that's the kind of logic or the kind of inference that you would draw from these sorts of cases. The other way of doing it is to temporarily disrupt the brain. So there are two methods that tend to be used. One is called TMS, or transcranial magnetic stimulation, and the other is called TDCS, or transcranial direct current stimulation. And the way that this works is that with TMS, you've got this, you can't really see the coil here, but it looks like a figure of 8, and it's got wires in it, and you pass a rapidly changing electrical current through those wires. It generates a magnetic field that's actually quite focused. It's about 1 centimetre squared. That magnetic field travels through the scalp and the skull, and it activates the neurons underneath it. So the neurons underneath, they fire, they generate their own electrical currents. What that kind of means is that if those neurons are firing in response to an external stimulus, they cannot fire as well or in synchrony with, say, normal cognition, so seeing a face or whatever it might be, okay? So you're effectively disrupting those neurons by making them fire to something that they shouldn't normally be firing to, i.e. an external current. Let me show you that.
I'm Roger Highfield. I'm the science editor of the Daily Telegraph. Now, words mean everything to me, but now I'm going to have the speech area of my brain turned off with a giant magnet. Humpty Dumpty sat on a wall. Humpty Dumpty had a great pull. All the king's horses and all the king's men couldn't put Humpty together again. Humpty Dumpty sat on... Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again.
That may have looked like a strange party trick, but it's actually part of a serious research project conducted by Prof. Vince Walsh in a basement lab at the Institute of Cognitive Neuroscience in London. He's using electrical and magnetic brain stimulation to understand the most complex known object in the universe and how to treat conditions such as migraine and stroke. Just to show the power of the magnetic fields passing through my scalp, and that this wasn't a trick, this is what it did to our videotape, even though it was positioned a few meters away. It's hard to see because of the interference, but here I could still sing a nursery rhyme, even when he disabled my language centre, because singing depends on the other side of the brain.
Humpty Dumpty sat on the wall, Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again.
I felt
Okay, brilliant enough. Okay. So obviously this has some advantages. So one, they're often called virtual lesions, and they're not lesions at all. You're not damaging anything in the brain. The effect's temporary, so you can obviously move them around. So you've got your own within subject control. You can kind of put TMS there versus TMS here, so you know that it's affecting different circuits. You can also investigate the time course of cognition. So these pulses are about a millisecond or so in duration. Obviously, the effects they have on neurons might last for tens of milliseconds. It's not necessarily the case that the effects are over straight away. But you can also investigate how. Oh, sorry, I haven't got that in here. You can also investigate how different regions communicate with each other as well. So you can combine TMS with fMRI, for instance. So you could, if you disrupt your face recognition area, for instance, you can see how that affects activity in your frontal lobes. You do that by applying TMS over the region you're interested in, and then you put somebody in an MRI scanner, and you can look how those two regions talk to each other or not when it's disrupted. So again, you can integrate it with other kinds of methods. Okay, any questions about TMS? Has anybody taken part in a TMS study? Okay, yeah, a few of you. I think it's fine, but you know, so, it hurt.
Yeah, it's like someone's flicking your head.
Yes. So what it can feel like is that so it doesn't just stimulate the nerves in your brain. It stimulates kind of the nerves in your kind of scalp or your jaw and so on. So particularly with the speech, if you're not careful, you know, some of these effects are not done by stimulating the brain. It's just your jaw muscle that go like this or whatever. Or the twitching, it might not be the brain. It might be your scalp that's been stimulated. Okay. Right, so it's quite late, but I'm happy to hang around and take questions for 5-10 minutes at the front. And next week we'll talk about the evolution of culture and social intelligence.